First off, let's do an overview of the cell cycle. To begin the cell cycle, it starts with the G1 phase. This is the phase of cell growth. The next area is the S phase, which is where the DNA is replicated. Down here at the bottom is the G2 phase, and this is when the cell begins to prepare for mitosis. Over here, this whole area is called the M phase. As a part of the M phase, mitosis and cytokinesis both occur here. To break down mitosis a little bit, there are four main steps, which include prophase, metaphase, anaphase, and telophase. This whole area right here includes cell division, and the whole process is interphase. Now, to go a little more in depth, here at the G1 phase, this is where the cells increase in size and they synthesize new proteins and organelles. Here at the S phase, this is where the DNA is synthesized and the chromosomes are replicated. Down in the G2 phase, this is where organelles and molecules that are required for the cell division, they are produced here. In M phase, there are two parts, mitosis and cytokinesis. In mitosis, this is the division of the cell nucleus, and cytokinesis is the division of the cytoplasm. Now let's go in depth about mitosis. Now let's go over four basic definitions that you need to know. First off, centrioles. These are tiny structures in cytoplasm that help to organize the spindle. The spindle is a fan-like microtubule structure that helps to separate the chromatids. The chromatids are each strand of duplicated chromosomes, and the centromere is an area where each pair of chromatids is joined. This is the whole cycle of the M phase. Prophase, metaphase, anaphase, and telophase are the four cycles of mitosis. To start off, let's talk about interphase. Prophase is the first and longest phase of mitosis. Here, the spindles are beginning to form. These are the chromosomes, which are the paired chromatids. Here is the centromere, which remember is where the two chromatids are joined. During prophase, the chromosomes become visible. The centrioles separates and move to opposite sides of the nucleus. The chromatin condenses into chromosomes and they become attached to fibers in the spindle. The chromosomes coil more tightly and the nuclear envelope begins to break down. Metaphase is the second step of mitosis. The chromosomes line up across the center of the cell. Each chromosome is connected to a spindle fiber at its centromere. Anaphase is the third phase of mitosis. Right here, these are the individual chromosomes. During anaphase, the centromeres that join sister chromatids split, allowing the central chromatids to separate and become these individual chromosomes. The chromosomes will continue to move until they are separated into two groups near the poles of the spindle. Anaphase will end when the chromosomes stop moving. Telophase is the fourth process of mitosis. During this process, the chromosomes begin to disperse into a tangle of dense material. A nuclear envelope reforms around each cluster of chromosomes. The spindle begins to break apart, and a nucleus becomes visible in each daughter nucleus. Mitosis is now complete. However, the cell division is not complete. Now that mitosis is complete, cytokinesis will occur. Cytokinesis is the division of cytoplasm, and this usually occurs at the same time as telophase. During this process, each daughter cell now has an identical set of duplicate chromosomes.